really excited to be here. I first came to the Sea Lab in 2019 for a workshop that Ronnie was running for surf and had a wonderful experience and met a lot of awesome scientists and collaborators. And so it's just really great to be back here and seeing everybody. So I'm really excited today to talk about some of the work that's going on in my lab. I just started in August at Villanova, and so I'm starting to build a new research program. So a lot of this is talking about things from my postdoc and things I'll be expanding on in the future. So my research, as Ronnie mentioned, is really focused on this idea of habitat utilization and consumer resource interactions, and how different environmental processes and anthropogenic impacts are influencing those across large spatial scales. And ultimately, I'm really interested in how these different utilization and resource interactions are going to affect overall ecosystem structure and function of estuarine and coastal ecosystems. And most of my work focuses on how habitats or foundation species are influencing energy flow dynamics and those trophic relationships. So I'm sure many of you in this room know that biogenic foundation species such as seagrasses, salt marshes, and oyster reefs are really important for a variety of ecological functions and ecosystem services that we as humans like. But the one I'm most interested in, and I think is one of the more important ones, is the provisioning of habitat for a variety of fish and invertebrate species, including economically important species such as red drum or blue crab here. But while we know that they're important for fish and invertebrate species, actually quantifying the relationships between a given habitat and a fish or the production of fisheries is really difficult. And so we have a few really good quantitative examples where we know X amount of habitat relates to X amount of fisheries production. And in this paper by Turner in 1977, this is one of the really few good quantitative examples of directly connecting salt marsh habitat to fisheries production. So here, as you had an increase in the amount of vegetated estuary, you saw an increase in shrimp production. But as I mentioned, this is really difficult to quantify. And there are a few reasons that these relationships are difficult to identify. So the first one is that there are intra-habitat gradients or landscapes or seascapes where we have a variety of different habitats within a given area. And so trying to figure out which one is directly contributing to fisheries production can be difficult. The second is that there's individual specialization. So fish and invertebrates, or nekton, can utilize different habitats at different points in their life cycle or different points in the day. So here by Roundtree and Abel, they kind of described these different patterns of habitat use by estuarine nekton. So we have our resident species like mummichogs that are utilizing a single habitat pretty much throughout their life cycle. However, we have ontogenetic migrants where a different species at a juvenile stage might be using a different habitat as an adult or a juvenile. And then we also have just cyclical migrants that are utilizing them at different time scales, whether that's over the course of a season, a tidal cycle, or over the day. I'm sure you guys know this too, but habitats are stationary, while many fish and invertebrates are mobile and moving throughout the estuary. So actually trying to identify when it's in this habitat and when it's getting that energy is really complex. And I love showing this figure. So this is data from Matt Kenworthy, who was my lab mate in graduate school. And so he acoustically tagged red drum in the Rachel Carson National Estuarine Research Reserve in North Carolina. And he was trying to quantify how much the red drum were utilizing different habitats. And this is just a really great visual depiction of how one fish over the course of a 3-hour period can use so many different habitat types. So you can see it's moving through this oyster reef and the mud flats into these seagrass beds. And towards the end of this video, you'll see it foraging or moving, we think is foraging, along the salt marsh boundary. And so again, how do you say this red drum is getting X amount of energy from this oyster reef when it's only spending a portion of its time there? And lastly, scaling up can be difficult. As I'm sure many of us in the room, as ecologists, we're looking in buckets or at small scale areas within the field. And so sometimes when you try to scale that up to a larger spatial scale, those patterns can flip or be non-intuitive. And so what I do for my research is I focus on these large scale or collaborative research studies that can reveal patterns across larger spatial scales. So for instance, in Whalen et al. 2020, we went to 25 seagrass beds around the world, and we were able to quantify consumption rates in all these different beds. And if you had done this just at one location, you might say, up in the north, you might say, oh, hey, consumption rates are fairly low. And if you go towards the southern parts of this latitudinal range, oh, consumption rates are really low, which is counterintuitive for tropical areas. But what we found by having this large collaborative network is that there was this mid-latitude peak in consumption rates for these species utilizing the seagrass bed. And lastly, the big thing I think it's interesting is these fish habitat relationships are going to be shifting across time, whether that's with these environmental conditions, say temperature or salinity gradients, or with human activities such as increased pollution and other variables. And so my research really focuses on some major overarching questions. So the first one, the main one, is how do intertidal habitats support these nektons, so those fish and invertebrates, and overall estuarine secondary production? And to tease that apart a little bit more, what variables, so what environmental or human action are influencing access to the habitats by these nekton, consumption rates and energy flow dynamics, and then how does spatial scale influence what variables are most important in driving these patterns? So today we're going to talk through two different larger projects that my lab is doing. So we'll talk about a few little mini projects within each one. The first one is looking at collaborative research to assess oyster reef habitat across the South Atlantic Bight. And then the second will go more into drivers of trophic interactions and energy flow dynamics in salt marsh ecosystems. So when we're thinking about quantifying habitat provisioning in coastal ecosystems, a lot of times people think about coral reefs or these beautiful clear water systems where you can just go out and count all the fish that are utilizing a specific habitat. So here we have our fish. We know they're directly interacting with that habitat. But as many of you know, working in Alabama, this is what our systems tend to look like in these intertidal coastal systems along the South Atlantic and the Gulf of Mexico. So how do I quantify if a fish is utilizing this habitat? It's pretty difficult. And intertidal oyster reefs are a great system to try to understand this because they're flooded throughout the day. We know they're important for fish and invertebrates. There are plenty of studies that show there are more fish and invertebrates on reef habitat compared to controls, like this example from Grabowski et al. And so that's great, but how does that vary across scales? And we know that traditional sampling techniques are really time intensive and often destructive to the habitat. So here we were just trying to take out a quadrat to count our oysters, and we saw a really big decimation to the oyster reef habitat. And so that's affecting how fish and invertebrates might use it. And so we really need to try to identify different ways to quantify fish and invertebrate use in these systems. We also know that it can be very labor intensive. You need a lot of people out there to put out these nets and count all the fish that are caught. And you're still destroying the habitat as well, just walking on that reef system. So the project that I worked on with a lot of collaborators was a National Estuarine Research Reserve Catalyst Grant. So this was a one-year grant where we were trying to assess how different tools and potentially non-invasive metrics to assess fish use of these oyster reef habitats was utilizable across this larger biogeographic scale. And we had a lot of different aspects of this project, from the habitat sampling itself to trying to quantify fish and invertebrates using different tools, such as eDNA, underwater sonar imagery, and traditional nets and traps. And then we also looked at qPCR to look at oyster disease dynamics. But today, I'm going to really focus in on comparing direct capture of these nekton on the reefs with our nets and traps to an underwater imagery, so that high resolution acoustic imagery. And then I'll also touch on some of our stable isotope results. And as I mentioned, this was a collaborative effort. So we worked with 5 different universities. This is when I was a postdoc at the University of Georgia. And we worked with this collaborative group to sample 4 national estuarine research reserves over the course of 2 weeks. And so we went out and sampled and were able to do a lot of really cool stuff. At each of these reserves, North Carolina, North Inlet, Sapelo Island, and GTM, we sampled 3 different reefs that were all approximately 1 kilometer apart. But we tried to focus on reefs that were characteristic of the natural reefs in these environments. And across this scale, we know that there are different environmental factors that can drive patterns of habitat use. And one of the key ones we know is the depth of water inundation over the reef habitat and how that varies across the scale. So we know in Georgia and in South Carolina at North Inlet, we're going to have higher tidal inundation and more water over the reef habitat. And so that might shift what we're seeing utilizing that reef. So we went out and we used pike nets to capture all the fish and invertebrates on the reef. We put these out at mid-tide in the evening and collect them the next morning at a low. So we ensured that there was at least 1 tidal cycle going through to catch everything that was utilizing that reef habitat. And this traditional nekton sampling provides really good insight to not only catch rates and abundance of fish on these oyster reefs, but also the nekton community, so we can look at community structure. So this is a unique aspect that we can't get with some of our other metrics. And so, for example, here we have our North Carolina NER. We see that pinfish are the most dominant species, which is not surprising if you've ever worked in the estuaries of North Carolina. But over in Florida, we mostly got silverside and some white shrimp. So there is some variability in just who's utilizing these different habitats across these sites. And in addition, we wanted to compare our traditional capture to our underwater sonar imagery or our high resolution acoustic imagery. And so we used an ARIS to do this. So those of you who are not familiar with this, the ARIS is basically underwater sonar that takes a beam down onto the reef and you get a reflection back. And so you can actually record fish and the movement of these individuals on the reef. So this is how we set ours up. We had our boat floating off from the reef. We had it at a 45-degree angle so we could get kind of on top and also down. And we were over the subtidal channel. And so that allowed us to see anything that was moving in front of the reef, but also directly on top of the reef. So this is what this looks like if we were looking top down. So we have our ARIS with our view field and we have our reef. So here we have our boat. This is where the ARIS is. Here's the reef. And here are some fish utilizing that habitat. So we went through and we actually had processed all of these videos and were able to quantify how many fish and get size structure of these fish on the reefs. So here's an example. So these are some videos of you guys can see of our unprocessed data. So we can see all these fish moving on the reef. Here we have a ray swimming across this oyster reef and hopefully foraging. So okay, this is great, but how do we process this? So what we do is we actually individually tag each fish that's in the imagery, and those lines that are flashing are us actually doing measurements of each individual fish. So we were able to actually get a really good number of what's utilizing the reef over just a short period of time. So these videos were about 12 minutes. So you'll see how many fish we got in 12 minutes in the thing. So just to show you again, this is the same image. This is our raw and our processed imagery. And you can see the fish school swimming across and us being able to get all this data. So what did we find? So when we compared the acoustic imagery to our direct capture, we actually found there was a really strong relationship between the abundance of fish we catch in the nets and the abundance of fish we see on the videos. But we see about 5 times more fish on the video imagery than we do from direct capture. And so while we're able to get a really good indicator in general of what's using the reef from traditional, this video imagery potentially could allow managers to visualize many more fish and get better numbers and indicator of what's happening on these oyster reefs. In addition, we do see that these two tools measure fish similar size structures on these reefs. About the ranges of the size structures are very similar. But in general, acoustic imagery is able to capture larger individuals than these traditional nets and traps from what we were using. Now, if we were using a gill net, we might have seen different things, but from these spike nets, which are a little less invasive, potentially keeping those fish alive, you're getting much smaller community structure or size structure. And then lastly, we wanted to look at stable isotope analysis on this reef. So stable isotope analysis will allow us to understand that next step of trying to get at function, not just the structure, but how are these reefs functioning as a food resource for the fish and invertebrates that are utilizing them. And so how do we use stable isotopes in ecology? So for this project, we focused on carbon, nitrogen, and sulfur isotopes to be able to assess different aspects of the food web. And it's really important because with stable isotopes, you are what you eat. You can see the different values and how they can correspond to the primary producers contributing to that energy flow dynamics. So just for example, the carbon source, their delta 13 carbon, allows us to understand what primary producer is getting eaten. So each individual producer has a unique value. Nitrogen is going to tell us something about the trophic level that the species is foraging at. So it tells us, are they a secondary consumer or a tertiary consumer? And then sulfur is actually really interesting. So sulfur allows us to better disentangle our carbon sources. So sometimes you can have overlap. So sometimes salt marsh and seagrass will have very similar carbon signatures, but they have very different sulfur concentrations because being a terrestrial versus a marine producer. And so this just allows us to disentangle this in isotopic space. So how do we collect stable isotope data? So we'll take our little fish here. our little minnow, and we take the tiniest little fish fillets. So we get muscle tissue, we dry it, we grind it up, and we run it through a mass spec to get our isotopic values. And I just want to highlight Eliana Fox. She was an undergrad who worked with me at the University of Georgia, and part of this project, she did a senior thesis focusing on mud crabs and how their resource use varied across these different sites. So she helped me process a lot of this data. And so if for those who have never seen a biplot before, here we have our carbon on the X for both of these figures. And then for one, we have our nitrogen, and the other, we have our sulfur. And what I want you to get from this is that these values are going to vary across species, but then also across sites. So we do see some clustering. So here we have our particulate organic matter, or what we're using as a proxy for phytoplankton. And we see they kind of cluster together by sites, but there is some separation in those values. We also see here, we have our Spartina on the other end, and we can see that our North Carolina samples have a much lower nitrogen value than others. So we're going to use these individual values to help autocorrect basically our trophic levels and our carbon sources. So we put all this data into these mixing models to get at resource contributions. So just looking at two species from the reef, the oysters and blue crabs, which are a dominant predator, we can see that there's some variability across sites in which primary producer is contributing the most. So we see for North Carolina, we see particulate organic matter, or phytoplankton, tends to have the highest contribution to the energy or secondary production of these species. Now, if we shift over to North Inlet, we see that there's actually a shift, and benthic algae, or microphytobenthos, tends to be contributing the most to oyster and blue crab production in these areas. We see the same for sapelo, although it's a little bit dampened. And then at GTM, we see that particulate organic matter, again, becomes really prominent and important in this food web. And so when we were thinking about this data, we wanted to know, okay, well, what potentially could be driving these? And as I mentioned before, we know tidal inundation is a very important driver of the differences in these sites. And so what we found is that tidal range is strongly correlated with whether benthic algae or phytoplankton is the dominant contribution of primary producer in the system. And so this is just one time period. This is just one site. But it's giving us a little bit of insight into what's going on these reefs and how food web structure might be different, or what producer is contributing is different across these large scales. So assessing reef conditions for fauna, basically what we found is there's no really easy way to do it, and using a combination of these different tools can really give us some insight into what's going on oyster reefs across the South Atlantic. So our ARIS and our nets, generate similar trends in abundance and somewhat similar size structures, although the ARIS is capturing larger individuals. We see isotopes are providing insight on that food web structure, and can potentially give us important information for management, focusing if we target sport fish or economically important species, understanding where they're getting those energy resources. And hopefully we can use these in conjunction to assess these communities over time and across reef types and across larger scale reefs. So for this project, as I mentioned, it was a one-year project, and so we're really hoping to target next steps. So there's another National Estuarine Research Reserve along this gradient where we're missing a gap in space, so we're hoping to incorporate them. And also seasonality, so thinking we only sampled in the fall, but do these trends hold if we sample in the summer when production's really high, or in the winter when there's very low habitat utilization? And we're also interested in exploring various reef types. So we had an end user workshop from this grant, and we talked to the different managers in the regions. And some of the things they're most interested in understanding is how does restoration and how does harvest affect what we're seeing on these reefs and how fish are utilizing them. And so going in and targeting natural reefs as well as harvested and recently restored reefs to understand how habitat utilization is changing over time. So next we're going to transition and we're going to talk about salt marshes, which is the bread and butter of the work that I do. I focus mostly in salt marsh systems. And we're really going to think about drivers of habitat access by predators, but focusing more on the trophic interactions that are occurring on the marsh edges, and also the energy flow dynamics. So again, thinking about stable isotopes and where organisms are getting their energy. And organisms, as I mentioned previously, move across ecological boundaries, and this can alter their trophic interactions. So one of the best examples of this that people think of are salmon. So salmon spend a majority of the time when they're growing foraging in the marine environment and getting these marine-derived energy resources. But as they get larger and they go up the rivers to spawn, they're actually transitioning that marine energy into terrestrial systems. So they move up into this area, some of them die, some of them get eaten by these bears or other terrestrial species, and then those terrestrial species defecate. And there's actually studies that show that these trees, hundreds of kilometers away from the marine system, have marine-derived energy resources in them. And so this is what we call a spatial subsidy. And I'm really interested in understanding spatial subsidy dynamics in marine systems. And so this idea, has been brought up in ecology and marine ecology in particular many, many times. So we talk about Teal and Odom's work on Sapelo Island, where they were looking at the flushing of energy and ecosystem dynamics, really some of the foundations of ecosystem ecology, where they saw marsh-derived energy is going offshore and enhancing these offshore communities. And then in the late 90s, Ron Knaib went out and said, hey, this is actually happening through a process called trophic relay or trophic transfer, where marine organisms, like marsh-derived organisms, are getting eaten by fish that live in the estuary, and then those guys migrate offshore and are getting eaten by bigger fish. And so that marsh energy is directly contributing to oceanic production. But again, as I've mentioned many times, quantifying that is really difficult. So how do we quantify that? Most people go from these small marsh-associated fish to smaller individuals, but what about big mobile transient fishes? And I think salt marshes are ideal locations to study this because they're at this land-sea interface, and so they have this more terrestrial-esque energy, and so it's easier to quantify in the marine system. We know that they're essential fish habitat for a variety of juveniles as nurseries to grow, and it's believed from Ron Knaib's work and Odom's work that show that the transfer of energy to estuarine species is really important. But there's some things going on. So there's some global variation in salt marsh habitats that are driven by a variety of different factors. And so most of this work where we looked at energy flow dynamics was at Sapelo Island, where there's really high tidal inundation and really high flushing rates. But so does this actually hold true? And does marsh energy contribute to estuarine species across other scales? And folks like Ronnie have looked at this, across the South Atlantic and the Gulf of Mexico, and found that there is differential amounts of energy that gets transferred to our species across these sites. And so just to highlight, some different sites where we have differences, there's differences in tidal inundation, there's differences in just the climate that a marsh is in, and this can alter a variety of things. We also know that there's differences in geomorphology or creek bank edges that can alter habitat use and access. And so a lot of these different variables are what drive some of my research questions. And so the main ones I really am focusing on is tidal forcing and vegetation, because we know from some earlier studies that they are really important in driving whether a predator can access these resources on the marsh platform. And we also know that tidal creek geomorphology can alter whether a fish or invertebrate is accessing that habitat and moving that energy offshore. So this study by Nelson et al. Up in Plum Island found that when they had really high amounts of human nutrient loading, that the edges of the marsh would completely calve off, and there became these like really large peat cracks and crevices. And when those occurred, the mummy trogs had much lower growth and production from the salt marsh than when they didn't have that and they could directly access it. So we know that geomorphology can alter this use and the trophic dynamics. But the drivers of marsh function may act differently based on the scale that you're examining them. So a recent paper came out in L&O Letters where we created a salt marsh typology. So trying to understand how can you characterize your marsh across the globe. And so we identified 4 different settings. We have the global setting, which is going to be driven by climate factors such as temperature and precipitation. Your coastal setting, which is going to be your tidal inundation, your wave energy. Are you a lagoon? Are you getting more riverine inputs? Your intersystem setting, which is where the marsh is occurring in the tidal position. So are you in the high marsh versus the low marsh? And adjacent systems, do you have oyster reefs? Do you have mangroves? And then lastly, your intrasystem. So that's going to be more of the landscape, the patch dynamics going on. And so my work has really focused on examining marsh interactions at 3 different scales. And when I was thinking about this, the typology didn't exist, so I'm trying to put it into a little bit of context. But we looked at the creek level, so that's going to be your intrasystem setting, the landscape level, which is a combination of inter and intraspecific, or intrasystem, and then regional and geographic. And so this is going to be more in our coastal setting. So I'm going to highlight 3 different little vignettes of looking at these different settings. So this one, we're looking at the creek level, and really we were focusing on how does the creek bank geomorphology, or the edge type, influence consumption of mud crabs on the marsh platform and also their prey density. And so this is work that was led by Grace Gibbs. She was a REU intern with me a couple summers ago on Sapelo Island. And she actually works at the Auburn Marine Extension right now. So yeah, this is some fun work that we did out on Sapelo. So what we did is we went out to a site on Sapelo Island called Dean Creek, where a lot of the Georgia Coastal Ecosystems LTER research is done. And we went along this single tidal creek, and we identified 3 different salt marsh edge morphologies. We had our natural sloping banks, which is just a natural slope that fish can just move up and access at the high tide. We had actively eroding slumps. So this is where the vegetation has fallen off into the tidal creek, but is still present. So there's vegetation sitting in the tidal creek and vegetation on the marsh. And this is really important because Georgia does not have any subaquatic vegetation. And so this is providing a new potential habitat for these aquatic organisms to access. And then we have our highly eroded escarpments. So this is basically, it's been eroded. There's a wall that and there's nothing else there. And so what we did is we put out some tethering assays. So as an ecologist, I like to glue things to a fishing line and see what eats it or how it gets eaten. So we sampled along this, we tethered mud crabs and looked at the relative consumption rates at these different edges. And what we found is that at these actively eroding slumps, there's significantly dampened consumption of these mud crabs. So almost a 20% decline in consumption at these different edges. So, but interestingly, we found that at our natural slopes and our highly eroded escarpments, consumption was pretty much the same. Mud crabs get eaten about 40% at both of those sites. But where we have that vegetation in the tidal creek, something's going on where the predators are not accessing the mud crabs on the marsh platform anymore. So that led us to this question. So if fewer mud crabs are consumed, does that mean there's more mud crab or prey density on the marsh, which could have a cyclical effect, right? So we have mud crabs that are burrowing, which could cause more erosion. So how does this play out? And what we found is that there's about 2 times as many mud crab burrows directly behind these slumps compared to on these other habitats. And I know mud crab burrows are a little questionable, but we're using them as a proxy for densities of mud crabs. And so really, this is really interesting in the fact that increased mud crabs potentially is causing that higher erosion, and more slumping is going to occur, reducing overall consumption rates on these tidal creeks over time. So overall, we found that habitat edge morphology is altering this cross-boundary consumption. We know that with climate change, previous studies have shown that there's going to be an increase in these slump rates. And so is that going to reduce the amount of marsh-derived energy that's going to contribute to these tidal creeks or to these fishes in the tidal creek? And so also at the landscape scale, does this creek bank structure affect our spatially coupled food web? So is it going to affect how much energy is coming from the marsh platform? So then we went and we said, okay, let's look at the landscape scale. So let's go to more than just one marsh habitat and see how differences in that landscape is going to affect energy flow dynamics. So specifically, we looked at the relative contribution of that salt marsh derived energy, so specifically Spartina alterniflora derived energy, to 2 highly abundant consumers. So we focused on grass shrimp and mummy chogs, which we know are resident in these tidal creeks. And then we wanted to understand how does resource use affect, or how does that affect resource use, the trophic position of these species, and their trophic niche, which is going to be an indicator of how many of the resources they're using that are actually available in the system. And so what we did is we were back on Sapelo Island. C is our Dean Creek, that site that we were just at. And then up here at B, we went to Bell Marsh. And we used a drone to assess the landscape structure of these in June 2022. Or 2023. I need to change that. It's 2023, not 2022. And so what we did is we went out and we used that drone imagery and we extracted elevation data as well. And what you can see right here very easily, Dean Creek has a pretty much one main channel, very few tidal creeks branching off of it. It's also a much higher elevation profile than our Belmarsh site. That's much more dendritic. The Spartina is existing lower in the tidal frame. And so you don't need to read this, but this graph is just, or this table is just showing you the difference. So the channel density goes from 13 to 73. That's a really massive difference. And our average elevation of the edge goes from 0.38 meters to negative 0.19 meters. So it's actually below sea level at Belmarsh. And so at each site, we collected 3 basal resources. We collected benthic algae, benthic microalgae, so specifically benthic diatoms, Spartina alterniflora, and then our suspended particulate organic matter. And then we had our mummy chogs and our grass shrimp. So to start with grass shrimp, what did we find? So we actually found that at both sites, there was similar resource use between these species. So the highest was our phytoplankton or suspended particulate organic matter. Although it was higher at Belmarsh compared to Dean Creek, it was still the most dominant source. Next, we saw Spartina, which contributed about the same amount at both sites. And then benthic algae was pretty low. Now, when we look at mummy chogs, there's actually something more interesting going on. So at Belmarsh, remember, this is our lower tidal elevation or the edge elevation and more dendritic system. We see that Spartina alterniflora energy contributes about 60% of the production for these mummy chogs, compared to at Dean Creek, where it's only about 40. And so we see a shift in the dominant resource being used at these sites. And I'm not going to show you guys the trophic position just for time, but there are differences in trophic position between the mummy chogs at both sites as well, but not for the grass shrimp. But what we found overall, looking at our niche, our trophic niche, we were able to see that there's much higher interspecific competition at Dean Creek in the resource use. So they're both trying to utilize phytoplankton and marsh, the Spartina about the same amount. And so we're seeing greater competition in the resource use of the grass shrimp and the mummy chogs compared to at Belmarsh, where they're actually having a differential use in their resource. So the mummy chogs are focusing more on that Spartina or that marsh-derived energy, while grass shrimp are focusing on the phytoplankton energy. And this could be due to life history strategy. So we know that mummy chogs are accessing the marsh platform daily. They're migrating on and off, while grass shrimp kind of just hang out in the same spot. They don't really move very much. They just want to hang out in their little spot and be happy. So at this creek level system, we found that landscape structure, specifically that marsh edge elevation and that channel density, play an important role in energy flow from the marshes. And the dynamics are also mediated by life history strategy. So again, now I'm thinking, let's scale this up. Let's go to a bigger scale. So at the regional or biogeographic scale, what variables are most influential in these food web diagrams? And so we specifically focus on how does tidal regime and habitat complexity across sites on a large scale affect consumption rates? And then how does that affect energy flow for a variety of mobile consumers? So thinking about larger mobile fish. And from a literature review that I worked on during my PhD, we know that the amount of marsh-derived prey in the diets of these big transient fish varies, and it has to do with tidal range. So once you hit a 1 meter tidal range, all of a sudden you start seeing these fiddler crabs and these mummy chogs and these marsh periwinkles showing up in the diets of these big fish. But you don't really see that when it's below that 1 meter tidal range. And so we knew tide was really important, also from other work that's been done across tidal gradients. So what we did is we wanted to test this across a smaller scale gradient. So North Carolina, where I did my PhD, is a really great natural laboratory to look at variation in tidal inundation, because it encompasses about a 15 centimeter tide all the way up to about a 1.5 meter tide across the state. And so we were able to go out to different marshes and set up these experiments where we, again, tethered some lovely little snails and looked at relative rates of consumption across these sites. And so what we found is that when we looked at maximum water height and inundation time, that more snails were eaten when you had greater flooding duration and more water over the marsh platform, which is to be expected. But we had some confounding variables. So when we went to our different sites, we found that our highest tidal inundation had the lowest shoot density of plants and the tallest shoot heights. And it was inversely related, where when we saw very little consumption, there was really, really high shoot density and really low shoots. So we needed to disentangle the tidal inundation variable from the plant vegetation variables. So we went out to our intermediate tidal site and we manipulated shoot density. And so what we did is we either removed marsh grass from a plot, from the ambient, about 50% reduction in the shoot density. We left it at ambient, and then we increased it. So we used these wooden dowels as a proxy of Spartina shoots, and we put out little dried squid assays to look at relative consumption. And so here's a mud crab hanging out, eating some dried squid in our plot. And so we know that it was working. We knew that these guys were eating in these sites. He's gonna swim away. Okay. So at the levels we tested, shoot density didn't seem to matter. So we went out in both the fall and in the summer, and we saw no difference across our density treatments. So this is kind of indicating to us that it's probably tide that's driving this, but we still needed to test shoot height. So we went out and we gave the salt marsh a haircut. So we went to our ambient sites, and then we trimmed the marsh down to 75 centimeters, 50 centimeters, 25 centimeters, and 10 centimeters, and we put out our snails again, because we know the snails like to move up and down the chutes, so this could be a big driver. And this was run by Mariana McMeans, who was an undergraduate at UNC who worked on this with me. And so what we found is that there was no statistical difference between any of our shoot height treatments and the relative consumption rate. But there was about 2.5 times more consumption in our really low shoot height treatments. And so potentially there might be something going on there that might be an ecologically relevant difference, although it's not statistically distinct. So basically from this, what we concluded is tide is more important in driving these consumption patterns than the habitat structure or the plant structure itself. And so that really got me thinking about, okay, but what does that mean for energy flow dynamics? So what we did is we went out to 4 different sites in Louisiana, North Carolina, New Jersey, and Massachusetts that have very different tidal regimes. And we collected a variety of different fish, invertebrates, and primary producer sources to run a stable isotope analysis. And I'm not going to go through all the details of that again, but what we did is we used our mixing models to look at the source use by multiple very mobile taxa. So we have a few marsh-associated species. So we have our killifish and blue crabs. These are going to be on the left. And then we have our more mobile taxa across the x-axis. And they're not going to be the same. We didn't have every single fish or species in every single site, but we tried to get ones that were similar. And then we have our source contributions. So to start, in Louisiana and North Carolina, what we found is that particulate organic matter or seagrass epiphytic algae, so more aquatic-based sources in blue, tended to have higher contributions to all of these species. And we do see that C4, salt marsh vegetation, so our Spartina, does contribute to things like killifish, and in North Carolina, some of these bigger species like red drum, but there's some variability there. And then what we found in New Jersey and Massachusetts is that while there's some very big variation, right? So we see killifishes pretty much are dominated by that C4 salt marsh vegetation. We see that there is a reduction, but overall, these higher trophic level species tend to have higher contributions of that salt marsh derived energy in helping to contribute to their production. And so it's just really important to note that we see these differences. And so we wanted to make sure that this was not due to trophic transfer efficiency, that the marsh production was able to transfer more efficiently in these northern regions, potentially due to colder water or things like that. And so what we did is we actually calculated trophic transfer efficiency, or the relative marsh production per consumer relative to trophic position. And so the slopes of these lines are your transfer efficiency. And basically there was no difference in transfer efficiency. So this indicates to us that the differences in the amount of production is due to the amount of marsh entering or energy entering the food web in those systems. So overall, what we found is that hydrodynamics and landscape structure can influence cross-boundary habitat use and energy flow dynamics. And we know that there's high spatial variability in this consumption and energy flow across both sites and scales, looking at all these studies combined. And so this is really what's driving a lot of my research questions moving forward as I start as an assistant professor and bringing in students to try to understand what's going on and hopefully broadening it to a new region up in the Delaware and Pennsylvania area. But again, this is really important to me because how is this habitat use and energy flow going to change in the future in response to these different multiple stressors? We already know that there is a lot of variation going on with sea level rise and human activities that are affecting our salt marsh ecosystems. So we really need to have this baseline information of understanding how it's contributing to our mobile fisheries and economically important species so we can predict how that might change moving into the future. And so with that, I have a lot of people to thank and a lot of funding sources, specifically the Gulf of Mexico Research Initiative that funded a lot of my PhD work, the NEAR collaborative system, as well as NSF